did in my group over the last 4 years or so, and as you see from the title, the main goal is to study to use the simulations to study the interactions of A beta peptides with the lipid bilayers, and this work really was actually done in jointly with Chris Lockhart. He's here as an audience. He, at that time, he was a graduate student. Now he's a postdoctoral associate in the group. So this is really kind of the work which we did together over these years. So first of all, introduction. So the A beta peptide itself is just a product of the proteolysis. This is a precursor protein. Unfortunately, I don't have a pointer. But this red section corresponds to the fragment of this precursor protein, which corresponds to the A beta peptide. In the APP, in this precursor protein, the peptide C-terminal of A beta peptide is partially embedded in the membrane. The rest is in extracellular environment. And when it is cleaved by several secretases from this protein, that's the sequence which you will have. And this sequence has two hydrophobic regions, this one and also in the C-terminal. But otherwise, I wouldn't say that it's sort of unusual sequence, and except that it doesn't form any well-defined native structure in aqueous environment. So this is the naturally unstructured, unfolded protein. Its, however, important property is that it readily aggregates and eventually form amyloid fibrils. So this is actually experimental structure. And this structure can be used as the definitive indicator that this particular brain was struck by Alzheimer's disease. So essentially, during the post-mortem examination, the formation of such fibrils is a strong indicator that Alzheimer's disease was present in this brain. However, the role, actually, the biological role of A beta peptide is highly controversial. It's probably as everything else in Alzheimer's disease. The more traditional point of view suggests that this is just the sort of incidental product of proteolysis, which happened to be cytotoxic. On the other hand, there is a kind of less mainstream approach, thinking that associating this peptide with the innate immune system, that this is actually antimicrobial peptide. In fact, actually, some studies, for example, done in Harvard, shows that this peptide is, at least against 8 bacteria, it's as cytotoxic as LL37, and this is a classical antimicrobial peptide. The role of A beta in the immune system response is sort of consistent with kind of controversial, I have to admit, but association of Alzheimer's disease with the either fungi or viral, for example, herpes type 2 virus infections. So it looks like that it could be that the A beta peptide plays an important role in the in normal sort of normal brain. And one way or another, without actually taking sides in this debate, the A beta peptides are highly cytotoxic. This is a lot of in vivo experiments which shows that the high degree of cytotoxicity of this peptide, and roughly the sketch of this process, and I'm not saying that this is a molecular mechanism, just a sketch, is that you have a beta peptide interacting with this lipid bilayer. It inserts deep into the bilayer, then it starts aggregate. What happens next, hard to say. Some experiments suggest that some sort of well-defined pores are formed as a result of this aggregation. So this is actually AFM, atomic force microscopy, tapping images of these structures. In the worst case scenario, when the conditions are right, actually these aggregated peptides can actually dissociate, physically disrupt the bilayer completely. So that means that the mechanism of cytotoxicity can be at least of 2 types. And I'm not saying that this is the whole spectrum, just at least 2 types. That there is some kind of formation of well-structured channels in the membrane, and I think according to my knowledge, probably the first who was articulated this was Arispe at NIH back in '94. Or it could be some kind of carpeting mechanism when the E-beta peptide, when it's bound to the bilayer, produce sufficient structural disorder on the lipid organization so that the, for example, ions can permeate uncontrollably through the bilayer. And not surprisingly, over the last, I don't know, 20, 30 years, there has been substantial amount of experiments done on the interactions of E-beta peptides with the bilayer. Generally speaking, looks like that under, for example, nanomolar concentrations, E-beta peptides interact with the bilayers as monomers. And by the way, physiological concentration of E-beta peptides is roughly in the picomolar range. By the way, so that means that individual monomers interact with this bilayer. And this is actually known very well from physics, from the biophysical studies, that usually binding of the peptide, absorption of the peptides on the plane just speeds up aggregation process, because instead of the three-dimensional bulk environment, you reduce your dimensions to 2 dimensions, and that essentially reduces the number of degrees of freedom. And also, again, multiple experiments shows that anionic bilayer seems to provide stronger binding affinity with respect to E-beta peptides. So this is a very, very sketchy kind of overview of experimental results. There is, of course, much more is known about this. But what my point here is that it looks like that, at least if you want to study the process computationally, probably the good starting point is to consider a single monomer interacting with the cellular bilayer. It's a kind of, it's a first step in the process of cytotoxic impact of this peptide. This is not to say that it is not possible that this peptide initially aggregates, and these aggregates bind to the bilayer later. This is another pathway. But based on the estimates of the concentrations, it looks like the monomeric binding is the preferable path. So where we are here, so we want to use molecular simulations to understand the molecular underpinnings of the interactions of E-beta peptides with the bilayers. And by saying this, I mean essentially several specific questions. First of all, I would like to know how the E-beta peptide changes when it binds to the bilayer. For example, in water, and this is, even though it is somewhat controversial, but the common point is that the E-beta peptide doesn't have well-defined structure in the water environment. How this structure changes when it binds to the bilayer. Second question is this: does it insert in the bilayer? And then, if it inserts in the bilayer, does it actually perturb the bilayer? What it does basically do with the bilayer structure and with the lipids there? And also, we try to understand how this process might be impacted by the third parties, by, for example, cholesterol, ions, and so on. So the approach is entirely 100% computational. It's not experimental, it's not theoretical, it's computational. So it means that it's, of course, we welcome experimental input, but our workhorse is our computers. And the second is that it's a reductionist approach. And you probably will not write this word in the NIH application. It's not fashionable. But I'm a strong proponent of this approach, nevertheless. Because what do I mean by the word reductionist? If you want to study the binding of the E-beta peptide with the bilayer, yes, of course, I would consider the E-beta peptide itself. Of course, I will consider the bilayer. Of course, I will have a water around it. But would I put all the different lipids which are present in a cell? Would I put, for example, initially, for example, cholesterol or ions or embedded proteins? No, I will not. I will simply consider the bilayer represented by the one type of lipids, for example, with ionic DMPC or anionic or particular type of lipids. This is, of course, simplification, obviously. But because currently the biophysical mechanism of interactions of these of E-beta peptides with the bilayer are not known on a molecular level, it's a reasonable first step in this process. And later on, sort of the degree of the reductionism can be reduced by adding more and more extra component. So what's the methodology? So here it is our simulation system in all its glory and complexity. So here you have every atom is represented explicitly in water, in lipids, so down to the individual hydrogens. And as a result, you have that many atoms. It's some people say it's large system, some people say it's small system, but one way or another, you need somehow to move these atoms around. So there are different methods in physics to do this. Some people can use Monte Carlo simulations. We use molecular dynamics simulations. And in the nutshell, molecular dynamics simulations are simple. I would say deceptively simple. You have the atom I, its acceleration times mass equals to the force. According to the theoretical mechanics, this force is related to the negative gradient of the potential energy. So here you have a link between the energetics and the movement of these atoms. The simplicity stops here. This is after that becomes only things become more and more complicated, because you need to integrate these essentially second order differential equations 30,000 of times at every integration step. This is extremely difficult computational problem. 30 years of research just barely dented, kind of make a dent in this, improve this efficiency. And let me say the following, that many people do this type of simulation. If you search, for example, biophysical journal or Journal of Molecular Biology or, for example, PNS, there will be a good deal of articles which are studying applying molecular dynamics simulations to this problem. What I think sets us apart is that we are trying to we apply additional algorithm designed to enhance conformational sampling, particularly this type of algorithm. And let me explain why you need this. So if you, for example, just take this structure and apply these equations, chances are that you will spend next maybe a month essentially just looking at this structure, because it will not change. The structure sits in the local free energy minima, surrounded by the large barriers, and it doesn't move. So if you want to connect these results with the experiments, you must have good statistics and good sort of distribution of structures which this peptide will sample. With the normal molecular dynamics, it's pretty much impossible task. However, the replica exchange sort of provide a possibility to overcome this problem. And very, very kind of briefly, just to give you a rough idea what this approach means. You do the following. Instead of considering just one system, you replicate the system multiple times. And for each replica, each copy of the system, you assign its own temperature. So for example, for the replica I, you will assign a temperature of interest, for example, 300 Kelvin. Physiological conditions. For all other replicas, you will assign the temperatures which is much higher than the physiological temperature. So you would say that why I need to have these high temperatures, because obviously it corresponds to some kind of unnatural membrane, unnatural environment. The reason is this, that when the temperatures are high, these type of structures can be easily interconverted. For example, this helix can dissolve, open up, so the peptide can unbind or bind deeper in the membrane. So it means that you have a diversity of conformations. And by cleverly designing the rules of exchange between the different replicas, exchanging the structures between different replicas, you can tremendously enhance the diversity of structures at the conditions of your interest, so for example, at physiological conditions. So that's essentially the idea of the replica exchange. It basically follows from the statistical mechanics. It's a well-established formalism, but as always, it happens in science. For example, I think that the first glimpse or ideas of this approach was first mentioned in '86 in the Phys.Ref letters. Then it takes another 10 years to recognize this in the kind of biomolecular simulations, and another 10 years to move more in the in the practical applicability of this algorithm. But the price which we pay for this is that it's computationally demanding, and that's why we need to use either Linux clusters or actually distributed computing. And by the way, this is the website which we use in partnership with the Parabon Computations, and you can visit this, for example, and see how you can use just the internet-connected computers for these simulations. Okay, but I think it's enough with methodology. Where's the sciences? So let me structure this in the following way, that I would use as a reference the properties of a beta peptide in water, which we studied years ago. I would consider two cases. One of them is the binding of a beta peptide to the anionic bilayer, DMPS. And another case is the binding of the beta peptide to the Zwitter ionic bilayer. And that will be kind of two contrasting cases, because one represents highly charged, negatively charged bilayer, and now one is Zwitter ionic bilayer. And the first question to ask here is how the structure, for example, secondary structure of the peptide changes when it binds to the bilayer. This can be done kind of seen in this picture. So this is the plot which shows the helical propensity for every residue in the peptide, and this propensity is computed by averaging over thousands, and I would say probably tens of thousands of different structures. And there is several sets of data. First of all, the helical profile in water, which basically shows it's consistent with the experiments that the helix is pretty much not seen in water environment. But when the peptide binds to either to the anionic or to Zwitter ionic bilayer, there is a tremendous enhancement of the helical structure. So for example, within the C terminal, this helix structure increases by a factor of more than a factor of 3 or more than a factor of 2 in the case of anionic bilayer. By the way, this result by itself is consistent with the experimental data. For example, 8 years ago, Graslund in Sweden, she measured the secondary structure of a beta peptide interacting with the SDS micelles, and the locations of these two helixes are matching very well our data. There is older work by David Craig as well in 98, I believe. So what about the tertiary structure? So if the secondary structure is affected by binding, and by the way, pretty much the effect of the anionic bilayer or Zwitter ionic is about the same. It's enhancement of helix. With respect to the tertiary structure, one possibility is kind of, if not to spread the attention across all possible contexts which the peptide can make, let's concentrate on the particular interaction. This is a salt bridge. It's the salt bridge formed between these two amino acids. And it is important interaction because, first of all, it is formed in the fibril conformation. And experiments suggest that, for example, locking these conformations enhances the fibrillization by about 1,000 times. So this is the important interactions for the, which is important for the aggregation of this peptide. So if you consider the probability distribution of the distance between these two amino acids for two different bilayers, you see a quite significant difference. In fact, actually, for the Zwitter ionic bilayer, this salt bridge is formed and it is disrupted for the anionic bilayer. It's consistent with this data. For example, in water, it's just barely present. Essentially, 15% is almost nothing. It's 80% probability formed in water, or completely, this actually was a remarkable result. It's literally zero in the DMPS environment. If you, for example, look at the radius of duration, and radius duration is essentially the measure of the compactness, of the size of the chain, of the peptide. Then in water, it's about 15. In the DMPS or DMPC environment, it's about 20. By the way, change of 30% in the radius of duration is a big change, by the way, because it's the average distance, typical distance between the pairs of atoms in the chain. So that's why, concluding, it looks like that the helical structure appears when the peptide binds to to any type of these bilayers, charged or uncharged. But the details of the tertiary interactions really depends on the particular charge state. So if it binds where the peptide is located in the membrane. So this is what simulations can offer for you. You can compute from the simulations the probability distribution of the location of the residue I at the distance z from the center of the bilayer. So this is the center of the bilayer. That's the distance z from this midplane of the bilayer. So the colors here, red indicates there is a high probability, blue low probability. So here you see that when the peptide binds to the DMP-C bilayer, there is actually a great deal of variability of the locations of a beta peptide. So especially the C terminal actually is located rather deep inside the bilayer. In fact, actually below these dashed lines, the dashed lines is the location of phosphate group. So it penetrates in the hydrophobic core, like for example, you see this in the snapshot from the simulations. This is the C terminal. It's this region which is inserts into the bilayer. For the DMPS, the situation actually is quite different. In fact, the peptide is just sitting most of the time on a surface without deep penetration. This is, for example, one of the snapshots from the simulations. And the same, kind of even more directly, can be mapped by the same observation can be made if you consider the density distribution of the peptide atoms as a function of z, as a function of the distance to the bilayer center. So here you have two, essentially kind of plot which has two panels, one of them for the zwitterionic and another one for the anionic bilayer. And the distributions of the essential distribution of the atoms, of peptide atoms. And they show that they're very different. For example, for the DMPS, it's a single peak. This single peak occurs at the distance more than 20 angstroms from the bilayer center, which means that it's consistent with this point which I made, that most amino acids simply bound, surface bound to the bilayer. But for the DMPC, situation is more complicated. You have one peak corresponding to the surface bound amino acids like those, but there is another subpopulation which actually goes in. So that to summarize, it means that the beta peptide looks to be not just binds, but also at least partially penetrates the zwitterionic bilayer, but it remains on the surface of the anionic bilayer. And initially it was not exactly clear why it happens, but after some kind of thinking, analysis, I think the explanation is the following. When the beta binds to the anionic bilayer, if you look, for example, at the strong interactions between the peptide and the bilayer, pretty much 80% of them are electrostatic. They are formed between the charged amino acids and charged lipid groups. But because charged lipid groups are on the surface, close to the surface, it's not surprising that the peptide tends to localize on the surface without going deeper. For the DMPC, situation is different. In fact, those strong electrostatic interactions, for example, between charged amino acids or, for example, phosphate groups and so on, forms just about roughly 10% of those interactions. As a result, the hydrophobic C terminal is free to go deeper. Most of interactions are hydrophobic or van der Waals interactions. So you have a very different picture or kind of interaction profile between these two cases. Okay, if the peptide embeds in the bilayer, does it do any damage to the bilayer? And again, from the simulations, you can compute this effect sort of directly. So this is the vertical cross-section of the bilayer. It's pretty much the same picture as this one, but we translate this picture into the distribution of the lipid atom density. So 0 corresponds to the middle position. So this is the midplane of the bilayer. This is the water below the lower leaflet, water above the leaflet. And again, Colors indicate high lipid density, for example, the red one, and the blue corresponds to low lipid density. And there's two variables. Z is the distance to the bilayer midplane, and the R is the distance to the center of the bound peptide. So far away from the bound peptide, you have kind of a normal bilayer. This is the where the most of the lipid atoms from the lower leaflet are located. This is from the upper leaflet. But within the A-beta binding footprint, you have the sort of the I would probably call it a density depletion vertex, something like this. So it's kind of a depletion where the region where the density of the lipids is reduced. And it is also seen that the surface of the bilayer, basically, to put this roughly, where the lipid starts as you go from large values of Z down to smaller values of Z, you see that there is an indentation. And this, by the way, the fact that the peptides make an indentation or thinning of the bilayer is a well-known effect, actually, from the experience. The question is by how much and what's the dependence of this effect on the properties of the bilayer. So this direct computation shows that for the DMPC, the degree of indentation is somewhat larger. It's about almost 3 angstroms. And that is consistent with the deeper insertion of the bilayer into the of the beta into the bilayer. But there is also two other impacts, differences between these two types of lipids. First of all, that when consideration of the density, surface density of the lipids, normally DMPS is far denser. It's far denser lipid bilayer. The area per lipid is smaller. The number density of lipids is larger. However, within this binding footprint, in fact, the situation is reversed. In fact, actually, the number density of the DMPS anionic lipids is even lower than of the DMPC, which essentially underscores that there is a much larger depletion of the lipid density within the binding footprint when you talk about the anionic bilayer compared to zwitter ionic. Second is the impact on the disorder, on the ordering of the lipids. So the typical quantity which is used in order to measure the degree of order of lipids, especially the fatty acid tails, is so-called SCD parameter. Without going too much into the definitions, let me say that this is the parameter which measures the orientation of the CH bonds in the fatty acid tails with respect to the normal to the bilayer. The larger the numerical value, the absolute value of this parameter, the higher degree of order. And here you have this parameter is computed for every carbon in the fatty acid tails. And there are two sets of data. In red, corresponding to the DMPC, to zwitter ionic bilayer, and in black for the DMPS, for the anionic bilayer. The fact that the anionic bilayer are more ordered is not surprising. This is consistent with the experiments showing that. Okay, let me turn it on. Consistent with the high degree of density of anionic lipids, and not surprisingly, because of this, you have a high degree of order. But what is important for us here is that you have two curves for example, for black data, and two sets of curves for red data. This is the upper curve corresponds to the this parameter computed for the lipids which are far away from A beta peptide, not affected by the binding A beta peptide, and this continuous curve in black corresponds to the peptide to the lipids which are affected by the A beta binding. So it means that it is the lipids which are within this binding footprint. So you see that the difference between the two is actually larger for the DMPS rather than for the DMPC, which sort of suggests that the degree of disorder, which is induced by the E-beta peptide on the anionic lipid is larger compared to the zwitterionic lipids. Okay, so if we kind of summarize this everything together, so essentially what type of mechanism we can envision or put forward based on this data? So here you have two bilayers. In black, it's the anionic DMPS bilayer. In red, it's the zwitterionic bilayer. So when the peptide binds to the zwitterionic bilayer, it utilizes mostly hydrophobic interactions, but to anionic, it's mostly electrostatic interactions. The peptide tends to be localized on the anionic bilayer, whereas zwitterionic, it penetrates deeper into its hydrophobic core. And then it looks like, and this is the results of the simulations, that the E-beta peptide tends to expel anionic lipids from its binding footprint. But it sort of tolerates and mixes well with the zwitterionic lipids. And as a result, it appears to be that the E-beta peptide disrupts the anionic structure to the larger extent than the zwitterionic bilayer. So this essentially picture can be illustrated with this plot. And this plot, it's a cross section of the bilayer. So here R is the distance to the midpoint of to the center of the bound peptide. The Z axis is the distance to the midplane in the bilayer. So this 0 corresponds to the midpoint in the bilayer. So this essentially, we're looking at the upper, let's say, upper half of the lipid structure. And these shaded areas represent the following. It's the areas where typically you find one particular molecular structural group. Based on the statistical analysis of simulation, essentially kind of circling the area where, statistically speaking, most of the time, particular, for example, type of structural group spends most of its time. For example, here now you use kind of purple probably color. So this shaded area corresponds to hydrophobic amino acids, location of hydrophobic amino acids. This one to the positively charged amino acid. This is to the negatively charged amino acid. This is the polar amino acids. This is the fatty acid tails, glycerol, head groups, and so on. So comparison of the DMPC, DMPS shows that these distributions of these molecular groups quite different. For example, for the DMPS, the very clear result here is that when the peptide binds, it has very well-defined hydrophobic core. And initially, we was kind of struck by this. We really didn't understand why it should be that way. But then, when you kind of summarize everything together, the situation sort of clarifies. Because here you have a hydrophobic core. That means that polar and especially charged amino acids are on the outer rim, on the boundary, around the peptide core. It is this amino acids which form the interface between the peptide and charged head groups. That's essentially implicating strong electrostatic interactions between the charged amino acids and the anionic bilayer. And as a result, because this is where the strong interactions with the bilayer are located, the peptide basically sits relatively high along the Z-axis. And its hydrophobic core just barely interacts with this fatty acid region, which is the hydrophobic core of the bilayer. But this is the location of the head groups of the fatty acid tails. This is the location of the head groups. So that means that if the head group is here, but the tail is here, it means that these lipids are highly tilted. They are not sitting in the normal undisturbed bilayer. They will be more or less vertical. Here they are highly tilted. That's, by the way, the reason why we're saying that there is a high degree of structural disruption of the disorder of the anionic bilayer. In the case of the zwitterionic bilayer, the situation is, as you see here, kind of messy. There is clearly no well-defined hydrophobic core. Instead, this hydrophobic amino acid tends to kind of push down and interact, and even partially, by the way, overlap with the fatty acid tails. And that simply underscores deeper insertion of this bilayer, lack of the well-defined hydrophobic core in the peptide, mixture of these amino acids, and also mixture with the lipids themselves. So you can, I'm not saying that this is a definitive sort of conclusion, but you can interpret this in the sense that the A-beta peptide tends to induce larger structural disruption on the anionic bilayers rather than on the zwitterionic bilayers. And no, we can, it's of course pure speculations, but we can speculate that because bacterial membranes tends to have anionic bilayers, anionic lipids in their bilayers, you can speculate that that might be the reason why A-beta peptide more active against the bacterial cells. Okay, very quick, just one slide, and this is sort of this promised decrease of the decrease of the reductionism. The inclusion of calcium ions. Calcium ions, actually, again, simulations on studying just calcium ions interacting with the bilayer are well known. There is no surprises here. It is known that calcium strongly binds to the anionic groups in the bilayer, particularly to the phosphate groups. And in a nutshell, our simulation suggests that when the calcium introduced as the third-party molecule to the system, it essentially just enhances binding of the A-beta peptide to the bilayer. And the mechanism of this enhancement, essentially of strengthening of the interactions between the peptide and the bilayer, is twofold. First of all, because the calcium ions binds to the zwitterionic bilayer, it basically makes it partially cationic. But the A-beta peptide is anionic. It carries negative net charge. So that means that it's not surprising that the interactions will be stronger between the peptide and the bilayer. Second effect is far more subtle and kind of not anticipated. That calcium ions actually also tends to change the tertiary structure of the peptide. For example, this salt bridge. This salt bridge is formed when you bind this peptide to zwitterionic bilayer in the absence of calcium. And essentially, it means that these two charged amino acids sort of are excluded from the interactions with the bilayer because they engage to themselves. Calcium breaks this bond completely, and essentially it frees these two amino acids, charged amino acids, for the interactions with the bilayer. That's another component here. Okay, then cholesterol, but this is a health-baked product. So this is unpublished data. The impact of cholesterol on the binding of A-beta peptides to the lipid bilayers is, again, as always, extremely controversial. If you look, for example, through the literature, there is opposite points of view, which says, point of view number one, cholesterol generally strengthens the bilayer. It's, for example, make it denser, reduces the area per molecule, increases the thickness, for example, of the bilayer. So from that point of view, binding is essentially, but to say not binding, penetration into the bilayer is discouraged because the bilayer becomes denser. As a result, the school of thought in the experimental community is that the cholesterol must offer protective effect against cytotoxic action. In our school of thought, which essentially just use different set of experiments, shows that yes, everything which you said is true, but on the other hand, this preferential binding to the surface, because the peptide cannot penetrate deep into the bilayer, speeds up aggregation. Aggregation creates more cytotoxic species. It disrupts eventually the bilayer from because the peptide adopts larger oligomers and it promotes oligomerization. So without taking sides, let's just only look at the what simulation says. Simulation says the following: that if you take the helical propensity of the A beta peptide binding to the bilayer, so that means that helical propensity for every amino acid. This data corresponds to water. I already showed this previously. The data, red data, corresponds to the binding of this peptide to the zwitterionic bilayer, which is cholesterol-free. Again, I showed this data before. The new set of data are in black. This is the binding of a beta peptide to the zwitterionic bilayer with cholesterol. 30, Chris, 30? 40. 40% of cholesterol. And you see here that helix is by and large disrupted. So it becomes comparable with water environment. Actually, helix, by the way, shifts somewhat. For example, the N-terminal is acquiring more helical structure. But by and large, the degree of helix which was seen, for example, in the bilayer without cholesterol is gone. Second is the degree of penetration of the A beta peptide in the cholesterol-rich bilayer. So again, this is the same plot which I showed before, except that there is two continuous lines here. The red one corresponds to the average position of each of these amino acids along the z-axis in the cholesterol-free bilayer. No, that basically shows that the C-terminal is partially embedded. For the computation for the cholesterol-rich bilayer, which is in black, shows that generally except maybe this position, generally the peptide tends to reside higher, more above the surface of the bilayer, which suggests that the peptide tends to be expelled from the bilayer. In fact, actually, Chris simulations shows that when we saw this first result, I said that, okay, maybe let's force the peptide deep into the bilayer by, for example, starting the simulations from the structure, which is completely sitting right in the middle of the bilayer structure. And the results of simulation shows that the peptide just is expelled from the bilayer. It goes to the surface again, and that suggests that the deep insertion of the peptide in this cholesterol-rich system is simply not favorable. Okay, why it happens, we don't know. It's work in progress. We need to figure it out, the molecular mechanism. For example, personally at the moment I don't understand why the helix is unstable here. We can only maybe speculate without benefits of the reliable data. For example, the formation of the helical structure in the case when there is no cholesterol can be traced to the fact that actually two factors. This region in the beta peptide has embedded helical propensity. Basically, there is some trend to form the helical structure if the environment is right. What the environment means? It means that, for example, if you can cross-link amino acids which are forming helical structure, and that cross-linking will be with the period of 4, because that's a typical alpha helical conformation. So, for example, if you can bring these amino acids with a period of 4 close together and stabilize their location by, for example, binding to the bilayer, you will stabilize this helical structure. This type of effect, for example, has been seen not just necessarily for the bilayer, but, for example, when the ibuprofen binds to this peptide. So this is apparently a relatively generic mechanism. It looks like that the cholesterol somehow destroys this. How exactly, we don't know. So that's the work in progress. So that's why, in summary, what I think I have 3 messages. So first is that I think I can show it. I did show that simulations, if you invest substantial amount of time into them and done correctly, can provide complementary description to the experimental data. It's not a substitution, obviously, of experiments, but it's a good additional tool, because I would say that it will be I know that maybe experimentalists have different point of view, but I suspect that it will be difficult to study the molecular mechanisms of EBT peptide interactions with the bilayer just using experimental tools. It's difficult to sort this out of all the interactions in play there. Second is that it looks like that overall, all of this work suggests that the EBT peptides should be probably more disruptive and more cytotoxic against the anionic bilayers rather than the heteronic one. And this is also the work on the cholesterol, which we do. And we also, which is not mentioned here, is that we trying essentially, as I mentioned, we want to reduce the degree of reductionism. And that means that, for example, Jim in my group is working on the binding of EBT peptides to the bilayers formed by two types of lipids. For example, anionic and zwitterionic. This is probably more realistic, actually, combination representation of the lipid. For instance, Amy in my group works on the actually even more complicated systems, which have free components, including also sphingomyelin and cholesterol. Niyati works on the unusual, actually, bilayer, which is formed by the mixture of lipids and lipopeptides. It's actually an unusual molecule which has the peptide head and fatty acid tail, and essentially kind of surfactant-like molecule, which can be also used for forming bilayer structures. So this is a work in progress. So in summary, funding, this is the recent publications, which covers the topics which I described. And thank you for my group. Without their work, this simply would not be possible.